So today's topics, they include the induced fit docking methods that are incorporated into ICM, the multiple 4D receptor docking, which is a method to incorporate an ensemble of receptor structures into your docking to represent flexibility or induced fit. I'm going to show you how to do explicit group docking, whereby you have grid maps for some of the region of your pocket, but you allow certain residues to be flexible during docking. And then just to introduce, there's no examples to show because they're quite long methods, but introduce you to the SCARE method, which is available in ICM, which involves dual Alanine Scanning and Refinement, which basically allows you to open up the pocket so you model the pocket. And finally, we'll be looking at the Libero method for generating an ensemble of pockets, as well as loop modeling in the region of the pocket, as well as normal modes analysis as well. So just if you're not aware of what induced fit is, what we're talking about today, so basically Emil Fischer's original theory about lock and key, the substrate and the receptor, is shown here, but the theory was elaborated upon by Daniel Koshland, whereby he suggested that maybe in actual fact the substrate and the receptor change together. Their shapes, they change their shape together when they join, when they bind. So we want to ideally be able to incorporate this in our docking. So these methods are going to show that. So the first approach we're going to talk about is the 4D multiple receptor conformation docking. So in this situation, you would have a ligand or ligands that you want to dock or screen, and then you may, for example, have 3 or 4 high-quality crystal structures of the pocket, and those crystal structures represent the flexibility of the pocket. So you may have one with a loop in, or a residue that comes in the pocket and out the pocket in each of the 3 conformations. So previously, before this 4D method was developed, you could do that in ICM. You could screen individually to each receptor shown here, independent runs, and then you would get the docking results, and you'd have to post-process those results, and you get the docking. It runs fine, but it's a very time-consuming approach, and to get to convergence using this way, it takes longer. So the 4D method is different in the fact that you take the 3 proteins, you superimpose those 3 proteins, and then you store the pre-calculated grids for each of the proteins of the receptors in one environment. And these are what we call the 4D grids, with the 4th dimension being the receptor. So there's a direct incorporation of discrete receptor flexibility in this approach. So the receptors can be provided by crystal structure or some models or other approaches to generate an ensemble of structures. And you need to choose an ensemble that represents the flexibility. So you ideally, you don't want too many, but you don't want too few, but you want enough to represent the flexibility of the pocket. And you can also use this to model waters as well. So you could have one receptor with the water in one position and the other with the water in the other. So basically the waters can be displaced using this method. So basically you put them all together, you superimpose them, and the docking is done in a single run. There's no multiple runs. And so the ligand is sampled using the Monte Carlo bias probability method, which we discussed in the first webinar. So it's randomly sampling the grids, all the grids for each of the receptors in one go. And the results are placed here. There's a published benchmark where they looked at 99 therapeutically relevant proteins and 300 diverse ligands, and 77% of the complexes were correctly reproduced. And the paper written by Giovanni Bottigoni is here, and this was done in Ruben Abergayan's lab at UCSD and Scripps, and Max Totroff here at MolSoft. So how do we do this practically in ICM? So in ICM, this is just I've just clicked on the icon and I loaded the ICM graphical user interface up. Here we have 3 protein structures from the estrogen-related receptor, which is an orphan receptor that acts as a constitutive activator of transcription. And it's an interesting receptor because it has a very interesting ligand binding domain, whereby if an agonist binds, then you can see this helix here, helix 12, comes across the pocket and closes in on the pocket. If an inverse agonist binds, this helix moves out, as can you see here. So and if we look inside the ligand binding pocket, you see there's lots of flexibility in terms of the residues as well. So it's an interesting test for the docking, for the 4D multiple receptor docking method. So we're going to do, basically how I set this up was I went to the search tab, I searched 1S9Q, which is the ERR receptor, searched 2GPP and 2EWP. You can see it has ICM written next to it on each side. So this means that I've converted each one to an ICM object. So if you remember from the first webinar, to convert, you just right-click. It would say XR here, and you'd see Convert ICM Object. You can go back to that webinar to see the different options that are available for that. But either way, we need this, in order to do docking, we need this to be an ICM object. And you can see that each one has has a ligand bound. This is the A one. It's actually not interested in the other molecules in each of these, so I'm just going to delete them just by right-clicking, selecting them, and deleting. Here, so now we're just left with the three structures. So as I mentioned, the most important thing is, well, one of the most important things when you're doing this 4D docking is that you superimpose the molecules correctly. So to superimpose, we need to select the A subunit of each one, go to Display tab, and go to Superimpose. I think I pre-superimposed them anyway. They're pretty good. The superposition option is pretty powerful. So basically, it's in the Display tab, and you click on the Display. So it superimposes whatever you have selected. So if you have, for example, let's find this region. Maybe we would be interested in just superimposing this region. So you just select that loop or that region, and then you go to the Display tab, and then you click on the Superimpose, and that superimposes it, just what you selected. So, but in this example, we're more interested in the superposition of the ligand binding pocket. Okay, so before we start, I think it'd be interesting just to have a look at the difference between the pockets of the inverse agonist and the agonist. The agonist structure is this one. So if we double click on here, we can go to Tools, 3D Predict, ICM Pocket Finder. It will run the ICM Pocket Finder, and it should find the agonist. This is the agonist pocket. The inverse agonist, we'll just run it for one of them. This is the inverse. This contains an inverse agonist for estrogen-related receptor gamma. And we go to Tools, 3D Predict, ICM Pocket Finder. Go OK. And we just double-click here, and we have the pocket. We have 3 pockets, but the one that's in the actual binding pocket is this one. I'm just going to color it a different color so we can see the difference. If you right-click on the pocket in the ICM workspace, and you can just change the color here. Okay, so you can quite clearly see there's flexibility in the pocket. There's difference. The blue one is the agonist pocket, and the red one is the is the inverse agonist. This change is primarily driven by phenylalanine 435, which we can just look at quickly. If we go here, if we expand, we can find the residue numbering, and the phenylalanine 435 is here. And if we select it, and we can display it in stick, and we can see that actually, you can see one here for the other inverse agonist, and the agonist one is sticking out this way. So this phenylalanine is particularly flexible, and it also controls that helix coming in and out of the pocket. So we have a decent flexibility example here. So to set this up for docking in ICM, first we need to the way it works is that we put all these structures, so we're going to place all of these structures into one single object, and I can show you how to do that. So we can place, we go to, because it has to be in one single object to dock, so it's going to automatically dock to each one, so we need to place it all into one. So what we're going to do is we're going to select a couple. First, actually, the most important thing is to remove the ligands out of the pocket. So we can see that we have a ligand here. We can just right-click and choose Move from Object. And the same with the agonist. The agonist ligand is here. You can just see it here. So right-click on it and choose Move from Object. Now you can see we have the ligands here, and they're outside of the receptor object. And the same with 2EWP. Right-click and choose Move from Object. Okay, so we now have these three ligands. We're not interested in those at the moment. We're more interested in the receptors. Okay, so we have the A chain of the 1s9q, the A chain of 2GPP, and the A chain of 2EWP. I'm just going to select the A chains of the two agonists, inverse agonists, shown here. And then we go to the mole mechanics menu, and we go impose conformations. So we're going to impose the conformations of these structures into this, into the agonist structure. So go to impose conformation. So the main object is the agonist, which is agonist structure, which is 2GPP. So we select it here. And the source objects are selected, so 2EWP and 1SQ9. So just go OK. At this point you can actually ask ICM to superimpose, but we did it using the display tab, or you can do it. And just for speed, I'm not going to re-optimize hydrogens. They're already done actually, so just go OK. So now you can see in our object 2GPP we have this new section called stack. So if I undisplay everything, and you can toggle the stack down, and you can see that we now have the structures in here. So this is 2GPP, this is 1SQ, it's 9Q, and this is 2EWP. So we have three. So you can add as many, you can add a number here, maybe up to 10 stack conformations for docking. Okay, so now docking is more or less exactly the same as before. So we're basically docking to this structure which contains the two other conformations. So to do that, we go to docking, and we choose New Project. We give it a name, so 4D. 4D. Yeah, you can't start with a number. That's worth. So doc 4D. Okay, and choose the directory that you want to save it in. Okay, so the receptor object is this one, 2GPP. So just choose 2GPP. And the binding site, we're going to define the binding site using this pocket that we calculated here for the agonist. But we've got to be careful when we do that, because we also want to incorporate the other agonist, inverse agonist site. So what we can do is we can use define site around this 2GPP one pocket. You can see the green crosses, and we can make that pocket bigger if needed in a minute. We can see that. In this example, we're going to uncheck make receptor maps because we're going to make 4D maps in a minute, so we don't need to do that. It would be a redundant thing to do. So as before, we have this probe, which is hard to see in this example, which basically means where the simulation is going to start. It's automatically placed in the middle of the pocket, and that's usually perfectly reasonable. And then you can so the next step is you go here, and you have the purple box, and you can change the size of the purple box. But I went too quickly. If you wanted to adjust that, you can make it smaller or larger depending on the size of your pocket. But I think we'll be fine in this example. And then to tell ICM to use the 4D approach to docking, we have to go to Docking menu. You go to Flexible Receptor, set up 4D grid. And it's asking you to remake the receptor maps. You can remake the maps. You have to remake them for the 4D approach. You go OK. I'm reluctant to do that because it takes a while to run. So you make the maps, and then what we do is just for this example, we're going to dock these ER agonists and inverse agonists into the pocket. So we can extract them into a chemical spreadsheet. So we have 3 of them. Remember, we extracted them from the PDBs. They've all got _1. You can view the structures clicking here. We're going to append them to a chemical table, and then we can dock from that chemical table. So we right-click on the ligand, and we say Extract Ligand as 2D drawing, and go OK. So it's placed it here, and we're going to add the other 2 ligands from the other 2 structures there as well. So you right-click on the next one, and go to Extract Ligand, append to the table called Chem, go OK. So now we have 2. And then the final one, we right-click here, also on the ATX. Which ones are we? This one, sorry, we didn't do this one. So right-click here, choose Extract Ligand, append to existing table, Chem. So these are the ones we're going to dock. So once the maps were made, we would go to Docking, and then Dock Chemical Table, and don't choose Dock immediately because we need to be careful. I'll show you that you'll see some. It's because the docking project is not set up that we haven't got OK to go. But I'm just going to show you the results now. So just going to close. So imagine we dock the table, and you'll get a dialog box telling you that the table is docked, and you'll get an additional table here called Chem Docked. These are the results. So this is the table we docked, and this is the panel we couldn't see because I didn't set up the project, but you can change the thoroughness of the docking to, especially when you're doing 4D docking, you may want to increase that to maybe 5 or 10, because you've got to have a longer simulation in order to sample those that ensemble of structures. So you can increase that to 10. And then you can also, this is the number of confirmations that it will report. So 3, it will return 3 confirmations. So then we went docking, we dock the chemical table. It runs. You get a message saying it's finished, and it will automatically build this hit list, or you can go to docking, make hit list here. But the hit list is made. And so as before, we have the score. So the lower the score, the better the prediction. So the score is related to the energy, the grid energy values for each of the terms shown here. So it's related here. And the final one is more interesting in the 4D docking because it's the rec confirmation. So it's saying this ligand binds best, gets this score when it's bound to receptor confirmation 3 in your stack. And you'll see when you, just going to clean this up a bit, it's displaying everything. So I actually called the project Dock 4D. And so this is the receptor in the docking. And you see in the docking, because we had a 4D docking, we had the stack, so you can toggle through those stacks. But when you click on the row in the hit list, it will take you to that conformation that it prefers for that score. You see for this one is 3, and it's 3. So this ligand is actually was taken from 2-EWP, and it redocks, and we can compare it to the crystal structure shown here. So the crystal structure is in green, and the dock one is in yellow, so it's pretty decent docking. Maybe a bit thinner. So for the second one, for example, as well. So this one should load up into receptors at the stack. The first one, it's saying here that it's from the first, it prefers the first member of the stack with this score. And if we can dock it there, the yellow one is the docked, and it's actually from the ligand to GPP, so we can just compare that with the crystal structure. And once again, just color the crystal structure one green. That's a pretty decent RMSD for those two. Okay, so that's the 4D docking. We discussed what the columns here last. If you want to, this for example, this is hydrogen bonding energy, hydrophobicity, van der Waals interactions, and so you can look into that into the previous webinars for those. Okay, so moving on. Right, so another approach might be, you may not have multiple receptor conformations, you may not be in that position, but you know from by looking at the residues that there may be certain residues that may be flexible, or you may want to allow one water molecule to be there and jump and not in certain conformations. So you can allow waters to jump to be present or not by using this approach called hybrid maps. So basically what you can do, we generate the docking project as before, but then you select certain residues that you want to be flexible. We don't recommend to select more than maybe 4, because you have to be very careful with the simulation, because you don't want to generate artifacts that will that will be incorrect. So just to be careful, you need to select 3 or 4 residues, and then, or you could just choose to select all hydroxyls, which will turn, and you can sample those, or you can choose water molecules. But yeah, this is one approach. So this is available in the docking menu as well as in the Ligand editor. I'm getting lots of questions, but I think I might have to cover them at the end. I apologize for that. Please feel free to keep writing questions, and I'll try to get to them at the end. So basically what we're doing, we have a grid, and residues are replaced by explicit atoms. So here we have an example in ICM. I'm just going to delete all. Go to File. These are all online, so you can reproduce these examples if you wanted to. So we have a kinase here. It's the fibroblast growth factor receptor kinase, and basically we imagine that at the moment we have this apo structure, and we want to dock to this structure. I have some FGFR inhibitors here, so we can basically we're going to dock it and see if it docks okay. If it doesn't dock okay, we need to incorporate some flexibility. So we go to docking. We say new project. We can say hybrid, dock hybrid, and then go. Actually, first I'm just going to quickly identify the binding pocket. So I'm going to go to Tools, 3D Predict, and then Binding Pocket, Isolate Pocket Finder, and it should find the ATP binding site. So it's got a table of hits. the ATP binding site is here. So we go to docking, new project this time, dock hybrid, and define sites around the pocket 2. Here you see green crosses. This is the pocket, the red one. And it's 1FGK2. Just go OK. So we go go. This is the probe. This is the box we missed last time. You can make it bigger and smaller by dragging on the corners. And just go go, and it will make the maps. Yeah, it will. I unchecked make maps. Just make them now. Okay, so we're going to dock an inhibitor to this pocket and see what happens. So basically we go to docking, dock chemical table. Chemical table is called chem. I don't want to dock immediately. You could do, but that's going to go okay. I just want to dock one of these just for speed. So I'm just going to uncheck this one, say it's already docked. So it means it's just going to dock the one that's not checked as docked. I'm just going to lower the thoroughness just for speed, but you may want to keep it as it is. And just go dock. So this may take, you can see that it's docking. It has what it will tell you at the top, one background job is docking. If you want to see the progress, you have a progress bar here, and also you can go to Windows menu, go to background jobs, and you can see that there's a process running here. You can right click and choose view output, and you can see the output place. This is what's going into the .ou file in your docking project directory. So it's getting there. It might take a little bit of time. Ah, it's done. Okay, so we get the hit list. And so the docking, you can see that I didn't get a score in this case because I didn't run with the scoring, but we can see if we undisplay the pocket that the lysine, that this molecule has a bad score. It doesn't dock correctly. And that's because in order for these inhibitors to dock, let me just show you, the inhibitor comes into this space here. And in the apo structure, this lysine is blocking the way. So this lysine, we can see if you superimpose many of these structures that this lysine comes in and out of the pocket. And so it's pretty flexible. So to tell ICM that it's flexible, and we want to keep this explicit during docking, so this will be a fully flexible residue, this lysine here. Let's just tag it just to make it clear. So so the lysine is here. So we go to docking menu, and we go to flexible receptor, and we choose explicit groups. So it asks us which residues do you want to be explicit. So we need to select that lysine again. There, you've got green crosses on the lysine. Just keeps toggling on and off. You see that the lysine is selected, and so just go OK. You could choose, we want to do full side chain. We want the full side chain to be flexible during docking, or if you check some other residues, you could just choose the hydroxyls and whatever waters that you have nearby to be flexible. And go OK, and then it will remake the maps with this removed from the map, and this will be flexible during docking. So I've run this, and I just have the results. So go File, Open, and this is so the scores are significantly higher now because we have with these inhibitors a score of -29. You can click on it, and you can see that when you click on each of the conformations, you can see that the lysine is moving out of the pocket, so it's been incorporated inside the docking. Okay. This is 2-FGI. You can see that so when you run this, you need to run with a thoroughness of maybe 5 or 10. When you do it from docking menu, docking chemical table, you can do it now, and you increase the thoroughness. And so you can see that the lysine is flexible as well as the ligand. Okay. So finally, just to show some other things, we have inbuilt into ICM. There is the scare method, which was published by Giovanni Bottagoni again. It's a new method for ligand docking to flexible receptors by dual alanine scanning and refinement. So it basically allows you to model the pocket and open up the pocket by replacing pairs of residues with alanine and then docking. So basically you have the receptor, you have the ligand. You do this dual alanine scanning where you systematically replace various pairs of side chains by alanines, and then you grid dock to these models with the alanines, and then you refine and see and cluster and see the score, and you see which conformation improves the ligand binding score. So it's a way of modeling the pocket. You can read the paper here. If you're interested in actually doing it, you can go to the help menu and online menu and go to the docking, use induced fit docking, and there's an example for scare. So the scare method allows you to take into account induced fit of the receptor upon ligand docking. So there's an example here. And another method for generating an ensemble of pockets for maybe 4D docking is the Alibero method. So this was published by Manuel Rueda and Max Totroff at MolSoft and Ruben Abagayan at UCSD. And the initial method is called Libero. It's a ligand-guided method, so generates an ensemble of receptor pockets. And so basically the first step in this method is that you generate multiple receptor conformations, and then you select those conformations that best perform in terms of being able to recognize actives from inactives. So this method is only useful if you have a set of data where you have known binders and some good decoys. So you can use those to take the 3D structures, you build an ensemble using normal mode analysis. Then you perform virtual screening with some Monte Carlo refinement. And then there's a fitness function to say how well the pocket that you generated using this actually discriminates binders from non-binders. If it's good, you can save that conformation. If not, you go back and repeat, and it runs like that. So you can use this in ICM. The scripts are actually hosted on GitHub, and they're very well supported by Manuel Rueda, who's now at Scripts, and you can download those scripts here. And the paper is here. So just wanted to show you other ways of generating multiple conformations for docking. So if you go to file, open. So one way to generate multiple conformations. So here we have a aldose reductase inhibitor, and let me just, these sticks are so thick. I know from looking at other structures that this loop here, shown in red, can come in and out of the pocket. So you may want to, if you have multiple crystal structures, obviously you could use the 4D docking, which we showed first of all, so you would read those structures in. But if you don't have multiple conformations, you can model, for example, this loop. So if we go to the, we first read in 1 PWM from search, and then we convert. So it would say XR here, you right click here, say convert, and then you see ICM. And it needs to be the current object. We say set the current. And we just choose the loop, which I know is here. You can see it's selected, green crosses here. It's also selected in the ICM workspace and highlighted in blue, that loop. Then you can go to Mol Mechanics, and there's some loop modeling protocols here. Go to sampling, and there's two options. Ideally you'd have time and you'd run this high precision sampling. So this does a full bias probability Monte Carlo simulation of the side chain and the backbone and everything, and it's a long simulation. And alternatively, you can download a database of loops from the PDB, which we have here, and then search for the PDB for search the PDB for loops that match this region that we're modeling, and that's a quick way of doing it. And I'm just going to just go OK and do that now. So it's going and it's comparing, looking for loops with similar distance and that we have in our database. And it's found, you can see here, it's found a stack of 88 different loop conformations. So you can expand that, and you can toggle through each one in the stack here. Okay, so for 4D docking, you may want to take some of the top 4 or 5 of the stack and delete the others. So you can select them, right click and say delete. Okay, yes. Then you have a smaller stack, and then you can just use this object as we did before. Go docking, new project, and then docking flexible receptor 4D grids using this object. And then you can dock chemical table or dock an SDF file that's saved on your machine in batch. Another approach to generate multiple conformations in ICM, as well as there's ways to you can run you can sample your protein or sample your peptide. Another way is to use normal modes, which is a method that uses like an oscillating system, a pattern of motion in which all parts of the system move with the same frequency and with a fixed phase relationship, like in a sinusoidal way. And so you can run. What you would read in your into your PDB, read in your PDB structure, convert it to an ICM object, and then you can run the normal modes method by going to Mol Mechanics, generate normal modes, and that too will generate a stack of conformations. So shown here, I ran it before earlier today, and you can see it generates a stack, and you can check. You could select 4 or 5 of those for docking, and this is what's used in the Alibero method as well. Okay. So just finally, just wanted to quickly show one thing that on the so far we've been using the docking menu, which is great for docking and screening. But if you're interested in optimization of your structures, we can. If you're interested in optimization of a ligand, you can use the 3D ligand editor to do that. And this was developed with Novartis, medicinal chemists at Novartis, and so you can edit the ligand in a single structure, or as this webinar is about 4D induced fit, you can also edit and optimize the ligand in using 4D as well. So to do that, we go to the ligand edit tab. We tell ICM this is the estrogen related receptor example we looked at before. We just choose which one we want to be the ligand we want to optimize, and then to set up the receptor in the ligand editor with 4D, we need to all we have to do is select the receptors that we want to include and go to protein. So go to this button here. This opens this setup receptor, and then choose here selection. So it knows it's going to use the 3 structures. And we need to make a box like before. You check the size of the purple box and go OK. And that makes the 4D maps, and then you can optimize your ligand using multiple receptors. We have a webinar on how to use this environment in a separate in a couple of weeks' time, I think. So just 1 second. So this gives you the panel to make modifications, and then you can toggle between the different conformations that you're using. You can look at the score in each one. You can make changes in 2D and 3D. We'll cover this in a couple of weeks' time. I just wanted to show you that it's available in the editor as well. And also in the editor you can allow, as we did before, you can define flexible receptor groups, so you can select residues and have those flexible like we did with the lysine in the kinase. And there's also side chain refinement here as well as in the docking menu. We got a new method actually for that, a new approach using the scare method, which we'll be releasing. So so the refinement is also kind of like so in the ligand editor you go to side chain refinement, and it would refine the side chains around your ligand. You can see that. Okay, so that's the end of the webinar. Just going to thank you very much for attending, and next week's webinar is on the covalent docking, and then we'll do fragment screening and linking, and then I'll say it's 3 weeks time in October, the ligand editor, and then atomic property fields, which is a 3D pharmacophore approach.